Good morning, everyone. Welcome to PMB 160, Plant Molecular Genetics. My name is Jen Fletcher, and I'll be one of your two instructors, along with my co-instructor, Bob Fisher, who is unfortunately out of town this week, but he will be back on Monday. We have two GSIs for the course, Brad Dotson and Yvonne Kim. Where's Brad? There's Brad. Yvonne is also out of town this week, but she'll be back on Monday. And we also have a GSI for the lab course, Stephen Yee, who is at the back. So I hope everyone has had a chance to pick up a copy of the syllabus and also a PDF of the first lecture. I'd just like to take a few minutes to go through some of the details of the syllabus and answer any questions you might have about it. Obviously, lectures Monday, Wednesday, Friday. There are two discussion sections, both of which meet Friday directly after class in GPB. We would appreciate it if you would attend your assigned section. If you, for any reason, need to change sections, please let Bob or myself know beforehand. We just want to try and maintain a balance of students within each section so you're getting an equal amount of attention. We have an office hour for Bob or myself. Every Wednesday after lecture, we can either do that here, or if we want to sit down and take more time, we also have a room assigned in 111 Koshland Hall, which is on the northwest side of campus. So you're welcome to attend any of the office hours or to communicate with either Bob or myself after class or by email. We have a website on BSpace, of course, which has the assigned readings. We'll also post the PDFs if you want to print them out in color yourself or just have an extra copy on file. We'll also have question sets and the answers to questions being posted onto BSpace. The recording of the audio, as long as I remember to turn the microphone on, and also hopefully a screencast will also be available through BSpace, and they're also, I believe, at least the podcast is also on iTunes. We're going to have a series of lectures on different topics. Some will be given by myself, some will be given by Bob. After each one of these sets of topics, there's going to be a homework set handed out. On average, I would say this is probably going to be once a week. There'll be a short answer type of format. You'll have one or two questions. It'll be worth 5 or 10 points. We'll assign that and give you a couple of class periods in order to hand it in. And then we'll make the questions and answers available on BSpace after the assignments have been turned in. As far as turning in assignments, you can either bring them to class. On the day the assignment is due, we'll have you bring them up to the front of the class after lecture, or you can also turn them in by email before the end of that lecture period. If you do turn them in by email, please turn them into your GSI and CC Bob and myself so we make sure that it's been received. Answers to homework sets that have been turned in after the answers have been posted to BSpace, which is typically right after the lecture where they're due, will not be accepted. Obviously, if you have a medical excuse, please let us know, preferably in advance if you can do it. In the past years, the total value of these question sets has been about 130 points, which is close to half of all the points available in the course. So it's really to your advantage to fill out your homework sets and turn them in, and it also gives you more consistent feedback on how you're doing in the course and how well your understanding is, rather than just waiting until the midterms and the finals. There'll be two midterms. They're scattered pretty well equidistantly through the course. Each one of them is 75 points. Then there'll be a final at the end of the course, which is 100 points. The final, unlike the two midterms, is comprehensive. It will be on all of the material in the course, but it will be sort of back-loaded towards the back third, on which you hadn't yet been examined. The laboratory course is a separate class on Mondays from 2 to 5. I think all of you who are in the laboratory course know about that and know what your assignment is. Then we've also, on the last 2 pages, given you the topics in the syllabus for each of the lectures and an assigned reading. And we do encourage you and expect you to do the assigned reading. Some of them are going to be primary literature review articles about a topic, which give you a broader overview than we can go into in class. Others will be primary research papers on a specific topic, so we can go more in depth in a particular topic than we can necessarily get to in lecture. Now, the material in the assigned readings are fair game for the homework sets. So just be aware of that. We won't ask you any direct questions on the exams, because that's a little unfair to ask you to remember specifics without being able to refer back to the text. But for the homework sets, you can expect questions about the readings. Any other questions in general about the business? Yes?
Online, it doesn't say what the textbook is.
there is no assigned textbook because we are trying to communicate material to you that in many ways is very, very up to date and changing quite rapidly. We haven't found that the textbooks can really keep up with that. So the assigned readings are really where you're going to find the material that complements the lecture most appropriately. If you're interested in a textbook that does cover a lot of this material, then I can recommend a book called Plant Biology, and I can put the details of that on BSpace if you're interested. Any other questions? Okay, well, let's go ahead and get started with the first set of topics. We're actually going to be looking at plant molecular genetics topically, sort of in reverse order. I'm going to start with genetics, and I know this is going to be a review for many of you, but I want to begin with this to put things into a context of how plant biology can be studied using genetic tools and sort of the fundamental principles that are applicable to plant genetics. Then Bob and I will talk in more detail about some of the tools and techniques and outcomes of molecular biology and genomics, giving you sort of a broader overview of modern tools that allow you to clone genes, perform mutagenesis, and then the remainder of the course, the bulk of the course, will be how we apply these molecular genetics principles and the molecular genetics tools in order to understand fundamental plant processes. And this is everything from development to hormone biology, to defense, to environmental interactions. So we'll see that a lot of what we understand about plant processes is really a result of experiments that have been done using molecular genetic tools. So the first reading will be from the classic source, Mechanisms of Mendelian Heredity, going all the way back to 1915. Pages 22 to 34 and 41 to 47 are the ones that are relevant. And note that these numbers are PDF file numbers, not page numbers from the book. So actually page 22 of the PDF file corresponds to chapter 1, page 1 of the Mechanisms book.
And you can find that on BSpace?
You can find that on BSpace. It's already been posted. You should have had an email notification about it in your mailbox. If you haven't, let us know and we'll make sure that you're actually on the BSpace site. We should have everybody's emails in there automatically, but it doesn't always capture everyone. All right, so as again, I said a lot of this is going to be a review, but I just wanted to touch on the key concepts in genetics that are going to be relevant particularly for this course. So we'll talk a little bit about genetic variation that we find within populations and the different traits that can be studied and have this type of variation embedded within them. We'll give a little bit of review on the storage of genetic information. That should be very basic, that you've already picked up in Bio 1A. We'll talk in more detail about the specific mechanisms of inheritance, which are really important for our course in understanding how we can apply genetic principles to understanding plant processes. And that leads to genetics as a tool then for the scientific study of heredity and of specific plant and also animal activities. So I want to just start with some definitions. Again, this is very basic, but just to bring us all up to speed. Organisms, as we know, whether they're plants, animals, fungi, archaea, organisms of all the different kingdoms consist of different traits. These traits are specific characteristics or features of the organism. So for plants, we can think of a whole laundry list: seed size, flower color, stem height, root length, lateral root size, number of lateral roots, sepal size, leaf number. All of these different things are different traits or specific individual features that each organism will have. But within and between organisms, there's some variation. So all of the traits of a given organism collectively are referred to as the phenotype. And these represent the expression of the genotype plus the contribution of the environment. So as we all know, even identical twins who are raised in separate environments are not completely identical. They have slightly different looks, slightly different behaviors, because there is a contribution of the specific environmental conditions in which they find themselves. But also we know there's a very strong contribution from the genotype, which is the specific genetic makeup of the individual at the level of the genes, and more specifically at the level of the DNA that composes the genes. Genetic information, of course, we know that DNA corresponds to nucleic acids that contain the genetic instructions specifying the development of all cellular life forms. Again, we find this in all three kingdoms. And specific genes are the basic units of that genetic information that determines the inherited characters. So the genes lead to the genotype. And of course, the genes consisting of the DNA are packed into chromosomes, the storage units of the genes, and there are thousands, literally, of genes that are packed into these chromosomes and compacted in order to be able to allow all this genetic material to get folded within the nuclei of a cell. And the genome, then, is the full collection of the genetic information, all the DNA and all the genes on all the chromosomes. And I want to kind of walk us back out to this very global level of thought about how we can analyze all of the genetic information at once, because in the past 5 to 10 years, new molecular and genetic techniques have been developed that allow us to, for any given individual or for many individuals, to actually assay the entire genome at once and how all of the genes are responding to a particular environmental condition or to a particular stimulus or to a particular stage or phase of development. And we're going to talk quite a bit more about this further along in the class. So zooming back down then into the chromosomes, here's a pair of homologous chromosomes. Of course, we know that you inherit one chromosome from the male parent, one chromosome from the female parent. These homologous chromosomes then pair with one another, so you have 2 copies of essentially each gene. And the locus is the name for the physical location of the gene on the paired chromosomes. So here would be locus A, locus B, locus C. In plants, particularly in Arabidopsis, each locus has a particular number designation, so it's unique and can be specifically identified as opposed to any of the other 26,000 genes in the genome. So the locus is the physical location. It's like the address of the house on a Google map. Because we have 2 chromosomes and they're copies of one another, they have 2 alleles, one copy of a gene at a particular locus. So at each locus, you have 2 alleles, one from each chromosome pair. Now, these alleles can be the same as one another, or they can be different from one another. So if they're identical, for example, you have a big A from one chromosome and a big A from another chromosome, or little a from one chromosome, little a from the other chromosome, or they can be different, where you might have big C from one chromosome and little c from another chromosome. What do these designate? They designate, of course, whether the gene is in a homozygous or a heterozygous state. So again, here we're just looking at 3 hypothetical loci on a hypothetical chromosome, where you can imagine for one gene at locus P, you have a big P from one parent and a big P from the other parent. So that's a homozygous condition. Each allele is the same. Here at locus B, or here at locus A, you could have small a and small a. This is also a homozygous state. Each one is the same. However, you can also have a heterozygous state where the 2 alleles for the traits are different. For example, big B and little b. This is a heterozygous state where the 2 alleles are different. So here we have an allele that's in a dominant state, and here we have an allele that's in a recessive state. Just to remind you what that means, a dominant allele is one that's expressed or one that provides a phenotype, something you can see as a trait, a visible trait, even when it's paired with a recessive allele. So for example, if we think of coat color, brown allele is dominant over the gray allele. So if you have a cat that has one copy of big B and another copy of big B, then the phenotype of this individual is going to be a cat with brown hair. However, if you have a heterozygous individual with big B and little b, which would correspond to gray, a copy of brown sort of cancels out the copy of gray, and you also end up with a phenotype of a brown-coated cat. A recessive allele is only visible when it's paired with another recessive allele, as in this example where you have little b and little b producing a phenotype of a gray-coated cat. Now, don't be confused by the fact that you've got 2 different names but only one allele designation. This is something that you'll find in plants. Typically, the mutant phenotype, the mutant phenotype confers the name, because we don't know what the normal gene is supposed to be. It's masked under wild-type conditions. In a mutant condition, you get a plant with small leaves, so you call it little SL, or you call it little s. But the recessive allele and the dominant allele will have different names, but only one will correspond to the abbreviation. Okay, so just don't get confused by, just follow whether the coat color or whatever the trait is dominant or recessive. The dominant allele is written as uppercase, the recessive allele is written as lowercase. Wild type versus mutant. This is a concept that, again, we're going to come up with many times in following lectures, where wild type refers to a genotype that's found in nature. What a wild plant species individual would look like, or a wild example of an allele. So an allele of a particular locus that's found in all of the wild examples of a species is the wild type allele, whatever that looks like. And it's often abbreviated either as WT, big or little, or as just plus. The mutant is a genetic variant of the wild type that usually confers a new type of trait. For example, in wild type plant species, most of them of peas, say, would be tall. But you might find a mutant that's dwarfed, much smaller size. So that would be something that's a genetic variant, different from the wild type. And that would be typically what would then give rise to the dwarf allele name. Small leaves is another example. So the mutant is the genetic variant, and the wild type is the normal correspondent. So I wanted to go back and go to the first principles of Mendelian genetics and just remind you that Mendel was an Augustinian monk in a monastery in Austria, what's now the Czech Republic. And that he was a teacher who was trained in math, statistics, physics, and probability, and was interested, in addition, in both plants and heredity. And fortunately for him, while he was assigned to teach other monks, he was also assigned to tend the gardens and grow the vegetables for the monks to eat. And one of the crop plants that he used was pea, because the mountains with their short, cool growing seasons made them an ideal crop to have lots of pea to eat. And so he also used pea as an experimental system. And by studying how different pea traits were inherited through the generations, he was able to come up with a number of landmark contributions about 150 years ago that we're still using today. That's the remarkable thing. So among his discoveries is he discovered the specific units of inheritance. And to set the stage, prior to 1860, people had thought that the offspring of two parents had the traits of the two parents blended together into the offspring. And this thought actually goes all the way back to Aristotle, who was spectacularly wrong about this and many other things. But you can actually understand that it's not correct just by looking, right? You can just look at the progeny of two people and say that's not two traits blended together. If you have two brown-haired parents and a red-haired child, you can say that that's not a blending of traits. But that was the prevailing paradigm. And what Mendel did was to say that's completely mistaken. Actually, there are specific units of inheritance that are determined by independent heritable contributions. So there's no blending that takes place. And this was a massive step forward. He also discovered that there were patterns of inheritance and the logic of this inheritance, and we'll talk about some of these familiar principles as we go along. He found that these heredity units, of course, he didn't know the concept of genes at that time, but he knew that they were maintained through the generations, even if sometimes that particular unit was hidden in one generation to another. Of course, we now know that these units are alleles of genes. He determined that one form of a gene, one allele, could be dominant to another. And he also discovered that the recessive allele is expressed only in the absence of a dominant allele, just what we went through a couple of slides ago. He also showed that alleles come from both parents. Of course, as you can imagine, back in the 1860s, the prevailing idea was that most of the information came from the male. Well, it turns out, of course, that that's not completely true. He formulated two very important principles that we'll talk about in more detail, the principle of segregation and the principle of independent assortment, in terms of this logic of how different traits are inherited. He published this work in a very obscure journal in the late 1860s, where it then sat and was completely ignored until the early 1900s, when two scientists, Hugo de Vries from Holland and Karl Correns from Germany, both did a similar set of experiments to what Mendel had done. And then, like any good scientist does, they went to the literature and they looked to see whether anyone else had come up with these same ideas. And they rediscovered these early papers by Mendel. and recognized them immediately as a landmark contribution to the understanding of how traits are inherited that we're still using today. And that's what's so amazing to me, is that all of these things that we're going to be talking about, principle of segregation, principle of independent assortment, how we actually study the inheritance of specific traits in an organism, is still something that I use in my laboratory today, and all other geneticists do as well, whether they're working on plants or animals or fungi or anything else. So Mendel's experiments were done with pea plants. They're essentially exactly the same thing as you would do in a laboratory today in pea or in maize or tomato. You would take 2 pea plants with different phenotypes for a trait. So in this example, we're looking at a flower color trait. We have one plant that has a white flower color and one plant that has a purple flower color. Now, peas, like many other plants, are actually capable of self-fertilization. They'll pollinate themselves and produce offspring by themselves without having to cross to another plant. So in order to prevent that from happening and actually do a cross, to look at how these 2 traits influence one another, what Mendel did was he took off the pollen from the the recipient plant before it matured, and then he actually physically took pollen from this white flower and put it on the carpel, the female reproductive organ of the purple flower. Then, as this plant was pollinated, the seeds matured into a pod. He then took each of the seeds in the pod, planted them, and looked at the first generation offspring to see what color their flowers were. Now, the blending hypothesis would say, okay, you've got a white flower and you've got a purple flower, so all of these first generation offspring should have, what, lavender, light purple, something like that. But what he found instead was all of the offspring had purple flowers. So now we can already see that the blending hypothesis is out the window. And in fact, Mendel actually studied 7 different traits, not just flower color, but also seed color, yellow versus green, seed shape, round versus wrinkled, plant height, tall versus dwarfed, and several other traits that I won't go into. And as of 2010, all 4 of these traits had been cloned molecularly. So we now know specifically the genes that are involved in specifying these traits. For example, there's a specific gene in the anthocyanin pathway that produces this purple color. When that gene is mutated, then you have white flowers because the anthocyanins can't produce that purple pigment. And anthocyanins are found not only in pea plants, but in many, many other plant species as well. So all of Mendel's experiments are still completely relevant today. So as far as the logic of inheritance, one of the experiments that Mendel did to understand how these units of inheritance are carried from generation to generation was to cross a set of true breeding purple flowers to a set of true breeding white flowers. This is called the parental generation, and it's denoted with a capital P. And again, we still use this nomenclature today. So if you're not familiar with it, please become so. He allowed these plants to cross to one another using that same technique that we just talked about. He then grew up the F1 generation. This is the first offspring. It's denoted with an F1. Sometimes the F is lower. This is the hybrid offspring of the parents. So now you have one allele from the purple flower parent, one allele from the white flower parent. But he observed that all the plants had purple flowers. So the white trait is now hidden. Now he allowed these F1 plants to self-fertilize. So this purple plant was crossed to itself, and then he looked at the F2 generation. And he looked at a large number of plants, and he found that of the 1,000 plants that he looked at, 705 of them had purple flowers, and 224 had white flowers. And that this was a ratio of about 3 purple to 1 white. So we've gone from the P0 to the F1 to the F2 generation. And now the purple trait has reappeared. So how can this be accounted for? Mendel deduced that there were discrete units of inheritance, what we now know as alleles of genes, that are passed on from each parent. He deduced that in this parental generation, the purple flowers must have had the unit big, what we'll call big P. And they had 2 copies of big P. Whereas the plants with the white flowers had 2 copies of little p, which would confer the white trait. When the unit from the purple flower was crossed to a unit with the white flower, you end up with purple flowers, but the designation is you have all of the flowers have one big P allele, one little p allele. Now these plants were allowed to cross themselves. They go through meiosis. In that process of meiosis, as you all remember, the chromosomes separate, and each gamete gets one or the other copy of each chromosome. Half of them will then get the big P, and half of them will get the little p. So big P, little p gets crossed to big P, little p. And in the ova, in the female, you have a big P and a little p allele. In the sperm, you have a big P and a little p allele. Then in the zygote, at some probability, you'll have one gamete with a big P joining with another gamete with a big P, one gamete with a little p joining with a gamete for the big P, a little p joining with a gamete for the big P, little p joining with the gamete for the little p. This is how you can get that 3-to-1 ratio. And this is the only way that you can get this 3-to-1 ratio, is by proposing this type of scenario. And by this, we can see then that the purple color is dominant, and the white color is recessive, because the heterozygous individuals that are big P, little p are purple and not white. If white was dominant, then what proportion of plants in this F2 generation would be white? 3, exactly, and 1/4 would be purple. So this principle of segregation is fundamental to this logic of inheritance, where the two units, as we now know, alleles of each trait segregate during the production of gametes. They go through meiosis. And the important point is that the segregation of alleles between the two gametes is random. It's not as if the male gametes all get the big P, or it's not as if all the gametes where you also have large flower size allele get the big P. It's completely random. So in the female gametes, 50% or 1/2 would have big P, 50% or 1/2 would have little p. And same for the male gametes, half big P, half little p. So then when these get brought together in the zygote, in the offspring, what you have is big P 1/2 times big P 1/2. So 1/4 of the total are homozygous big P. Here you have 1/4 big P, little p, 1/4 big P, little p, 1/4 times 1/4 is a half. Half of the offspring are heterozygous, big P, little p. And then the other quarter are homozygous, little p, little p. So half the gametes carry one allele, half the gametes carry the other, and they get assorted by this methodology. All right, so is everybody following this so far? Great. All right, now what's the square that we've organized here to arrange the different distributions of alleles? It's actually a really useful technique. The genetic problems, as we're going to see, can be readily solved using what's called a Punnett square. And that's what this is, and it can become more elaborated upon if you have more than one trait, as I'll show you in a little bit. It's a tool for calculating genetic probabilities that was named for this English geneticist called Reginald Punnett. So what you have is a square where for one trait, you put the female reproductive cells on this side, male reproductive cells on this side, and then at each location, you put what allele is coming for each gamete. So in the previous example, we had a big P and a little p for the female, big P and a little p for the male. And then you can solve the Punnett square just simply by combining the allele in each of the squares. So how do we use this Punnett square in principle? For example, let's say we're going to make a cross between two plants that we already know are heterozygous for an allele that causes dwarfism. So in this case, we're going to use D to designate dwarf. The dominant version is the wild type version, and that's the normal tall plant. The recessive is the dwarf, and that's little d. So these heterozygous individuals are going to be big D, little d at the genotype level. That's what their alleles look like. At the phenotype level, what the trait shows is that they're all tall. So we take this big D, little d individual, cross it to another big D, little d individual. And then we show what the gamete formation is going to be by the parents into the F1. This parent can contribute a big D to the gametes or a little d to the gametes. Remember, the probability is 50%. And same with this one. So these are our 4 gametes. So we set up the Punnett square. We put the male gametes on this side, big D, little d. Female gametes on the other side, big D, little d, or you can do it the other way around. It doesn't matter. Now we can solve the Punnett square by simply drawing in each of the alleles in each box. So big D, little d in this box, big D in this box, big D, little d in that box, big D, little d in that box, little d in that box. From this, we see the results are that you have 1 big D, and 2 big D, little d, to 1 little d, right? So that's what the genotype is. That's the distribution of the alleles in the offspring. The phenotype, then, is 3 of the individuals have the dominant allele, so they're going to be tall. 1/4 of the individuals have only the recessive alleles, so they're going to be dwarfed. So the genotype is 1 to 2 to 1, 1 homozygous wild type to 2 heterozygous to 1 homozygous mutant. The phenotype is 3 tall to 1 dwarf. So you can see it's very straightforward then to solve the genetic crosses using the Punnett square. And this is a hint, this may turn up on a homework set. So we can use this process then to make test crosses where you don't know what the parental genotype is. You can figure it out by making a test cross that, for example, looks like this. All right, you've got some purple pea flowers. You don't know if the plants are homozygous wild type or if they're heterozygous at that locus. So how do you figure it out? Well, a simple thing to do would be to cross it to some white pea plants that are recessive at that locus. So they're little p, little p. You make this cross, you look at the F1, and you can do the Punnett square. So you do 2 Punnett squares and see which one fits the data. If this parental plant was big B, big P, then you're going to find when you cross it to this white flower that from this parent you have big B, big P, from this parent you have little p, little p. You then solve the Punnett square. 100% are heterozygous, 100% are going to have purple flowers. But if your parental plant was heterozygous, then the alleles are different. Then you're going to have a big P and a little p from this parent, and a little p and a little p from that parent. Now you see that you get heterozygous from this combination, and you get homozygous mutant or homozygous white flower from the other combination. So if I were to ask you then, if you see an F1 population where 50% of the plants are heterozygous purple flower and 50% of the plants are homozygous for white flower, you could then tell me what the genotype of the parents was, right? So you do the test cross to see which cross fits the observation, and that allows you to conclude then what the parental genotype was. Now this is an example of what we call a monohybrid cross. It's looking at a cross to analyze one trait, but you can do this to analyze any number of traits. And so Mendel did a lot of experiments on 2 traits, dihybrid crosses, so plants that differed in 2 different characteristics. And this allowed him to study the inheritance of 2 different traits simultaneously. So for example, he would look at trait 1, seed shape, where he had populations of plants that had round seeds, populations of plants that had wrinkled seeds. And he would look at trait 2, seed color, yellow seeds versus green seeds. And he wanted to see what the distribution was of these different characteristics. And he made observations then to determine the pattern of inheritance for 2 traits at once. So here's an example of a cross in which the parents have yellow round seeds, parent 1. The other parent has green wrinkled seeds. Now this is another example of a P-run cross that will get the same outcome. Okay, we're not crossing this one to this one. These are 2 separate crosses. So just for the purposes of the lecture, let's ignore that one, but it comes out the same way. All right, so we do this cross. You take a yellow round seed individual, cross it to a green wrinkled seed individual. And you observe that in the F1, 100% of the plants have a phenotype of yellow round seeds. So we can already infer from this what? What's dominant? Great, yellow and round. So we take this yellow round seed individual, allow it to fertilize itself. So F1 being crossed F1. And we look at the distribution of traits in the F2. Now what we observe is a different set of outcomes. We find all 4 of the traits come back. We find that 9/16, so if we looked at 16 seeds, on average 9 of them would be round and yellow. 3/16 would be yellow and wrinkled. 3/16 would be green and round. And only 1/16 would be both green and wrinkled. So we can then look at a Punnett square to figure out how this distribution came about. So we've already determined that yellow and round are dominant. So green and wrinkled are recessive. So we're going to use G to refer to green, and we're going to use W to refer to wrinkled. So in our original cross, this parent that was green and wrinkled is homozygous recessive for the green allele and homozygous recessive for the wrinkled allele. The other parent, which was yellow and round, is homozygous dominant for the green allele, homozygous dominant for the wrinkled allele to produce yellow round seeds. Okay, so if we then look at the gamete formation in the F1 of this cross, the only gametes that can be produced by this parent are big G, big W. The only gametes that can be produced by this parent are little g and little w. These gametes are then fertilized, and all of the F1s, as we saw, are yellow and round. They're heterozygous at both alleles. Okay, so far so good? So now we're going to take these plants and allow them to self-fertilize. And then we now have a larger Punnett square because we have more different allele combinations. In the F1 cross, this parent can produce a big G and a big W, a big G and a little G and a big W, little g and a little w. And remember, we already know from the first principle of segregation that there's an equal probability that any gamete can get any of these different combinations. So it's 1 to 1 to 1 to 1. Same with this other parent, which we draw along the top. And then once again, you just fill in the Punnett square, very straightforwardly. So this individual has all homozygous dominant alleles, big G, big W, and produces yellow round seeds. You can just continue to look at each of the different combinations. And you can see here, for example, among gametes that have little g and little w, most of them are going to be receiving a homozygous dominant allele from the other parent, either both G and W to produce round and yellow seeds, big G, little w to produce yellow and wrinkled seeds. Now you've got 2 recessive alleles at that locus. Here you got little g producing mutant green seeds, big W, little w producing round seeds. And only this individual is going to have a recessive copy of all 4 alleles and produce green wrinkled seeds. And you can see that the number of boxes we have are 16. And that's how we can fit this to the observation of 9 to 3 to 3 to 1 out of 16 individuals. Here are the 9 that are yellow and round. 1, 2, 3, 4, 5, 6, 7, 8, 9. Here are the 3 that are yellow and wrinkled. 1, 2, 3. Here are the 3 that are green and round. 1, 2, 3, and 1 green and wrinkled. So the summary of this dihybrid cross, as we saw, is the F2 genotypic ratio. Looks like this. You have 1/16 that has all 4 dominant alleles, 2/16 that have 3 dominant, 2 big G, big W, little w, 2/16 big G, big W, little w, 4/16 that are heterozygous at both alleles, big G, big W, little w. Out of this, 4, 5, 6, 7, 8, 9 16ths have the phenotype of being yellow and round because they have the yellow trait being conferred by big G, they have the round trait being conferred by big W. You also see 1/16 that is homozygous big G, homozygous little w, and 2/16 that are heterozygous big G, little g, and little w, little w. So this is the 3/16 that are going to be yellow and wrinkled because 2 plus 1 has big G to confer the yellow trait, and all three of these are little w, conferring the wrinkle trait. Okay, we can continue with this. Here's the 1/16 that are heterozygous or homozygous mutant in both alleles, little g, little w, producing green and wrinkled. And this actually fit very, very well with the studies that Mendel actually did. So this is some of his data where he did exactly this cross of round yellow seeds to wrinkled green seed plants. In his F1, he observed all round and yellow seeds. In the F2 generation, he looked at several hundred plants, found that 315 had round yellow seeds, 101 had wrinkled yellow seeds, 108 had round green seeds, and only 32 had both wrinkled and green seeds. And if you look at the ratio, you find that it's a ratio of 9.8 round yellow to 3.2 wrinkled yellow to 3.4 round green to 1.0 wrinkled green, which is very, very close to that expected ratio of 9 to 3 to 3 to 1. So these are examples then of how we can use genetics as a tool for discovery when we look at inheritance patterns. I've already shown you that it's a way to predict traits. You can predict the offspring phenotype from the parental genotypes. If you know you've got two homozygous dominant plants, you cross them together, you're going to get homozygous dominant offspring. And as I showed you, you can determine parental genotype from the offspring genotypes. You can look at your F1 plants, see are they 100% purple flowers or are they 50% purple flowers, 50% white flowers. And based on that, you can then infer what the parent genotype was. And it's also a way to breed traits. You can plan specific crosses in order to obtain the desired phenotypes. And this is often where these dihybrid crosses come in, where if you want plants that have wrinkled green seeds, you can then figure out what cross is going to be required in order to obtain that desired outcome. So that's it. Are there any questions from today's lecture? Okay, I will see you Friday then, and we'll have the first homework set on Friday as well. Thank you.